In this video we will discuss about a new paper where they have discussed about cell to cell transfer via tunneling nanotube in pancreatic cancer cells from the macrophages. So we will start with the first figure that is presented in the paper, and in the panel A they have discussed or they have described how they have did their experiment. So in order to create a hypoxia condition, what they did is they co-culture cancer associated fibroblast with the normal fibroblast, and from this they collected some medium, this condition medium, without gemcitabine treatment, and another medium with gemcitabine treatment of 10 macromolar. Gemcitabine is a drug that is used to treat cancer. And in the second phase, what they have done parallelly is that they took their for the macrophages, they took the monocytes and differentiate themselves into the macrophages and co-culture them with the pancreatic cancer cells. And the pancreatic cancer cells are red fluorescent, are labeled as red fluorescence, and macrophages are labeled as green fluorescence. And thereafter, after co-culturing them, they separate out the cells that are double labels, or this differentially double fluorescent cells are being separated out, uh, so that they can know that these cells have now both the characteristics of the macrophages and the pancreatic cancer cells. So this is the overflow that they have used in this figure or for this experiment. So the panel B shows the cell images. Panel B shows the cell images just before they go for this flow cell sorting, that is at this stage, just before they go for this FACS. So in this figure they have showed that many these white arrows, and these white arrows are pointing towards this tunneling nanotube, what they called as, and this could be, and this is uh, this is considered as tunneling nanotube, which is used to transfer informations between the cells. That is, intracellular transport might have occurred through these tunneling nanotubes that are shown by the arrows. So, and this thing has already been published in a previous paper, so they have not discussed how this thing works in this paper. So when they did the cell sorting, so this is for the different kind of cells, and also these large cytoplasms. Large cytoplasm also indicates the cell intracellular transport that occurs between the cells, either between the cells, that is between pancreatic cancer cells and macrophages. And thereafter, once they did the cell sorting, in this they have find out a different kind of populations, and they did the cell sorting not only on the adherent cells but some on the dead cells also, which they have observed after seeding them in a co-culture medium. That is, FACS is done after on the third day. So in this they have find out three different populations. First one is the pancreatic cancer cells, which are red in color. Next one is this macrophages, which are green in color, and the third one is the doublets or double fluorescent cells, which are in this figure blue in color. And when they check the populations of the doublets in the adherent cells, they see that these are around 9.4% compared to the detached cells, in which they are around only 0.3%. So from this they indicates that the survival possibilities of the double fluorescent cells are higher when they are higher in the when they are in the adherent cells, or they are, we can say that the cells in which this intercellular transport occurs have a high possibility of survivals, since their populations in the detached cell is very low, whereas the pancreatic cancer cells and the macrophages in the detached cells are still shown at a higher level. And when they use When they use, apart from these hypoxia conditions, they also use normoxia condition. In the normoxia conditions with this gemcitabine, they can see that with the gemcitabine treatment, they can see that this doublet populations is around 12.9%. However, when they don't use any gemcitabine treatment, the population goes down. So this indicates that cell-cell transfers are more prominent when there is a some kind of stress conditions available. And the panel F is the confocal images at different Z conditions, indicates that some of the spots are completely green in color, and the author says that this are the pancreatic cells which has acquired the properties of the macrophages because of the intracellular transport. And that is the and this is also there we can also see that evidence. So this is not what the professor actually says, but when we see these images on the different z-axis, we can see that some of the green, this is not what the author says, but authors most focus is on this kind of images where we can see some green spot on the pancreatic cancer cells, indicating that the macrophages has acquired the pancreatic cancer cells have acquired some of the macrophages properties. That's why the pancreatic cancer cells has this green fluorescence. So this is what professor wants to not professor the author wants to convey from these experiments. So if you need to understand more about their experiment and this intercellular transport via tunneling nanotube or the other approaches, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.